Okay, so where we left off is the gates opening and closing at each node. And so the last thing we need to do is look at what happens at that last node to trigger what happens at the synaptic terminal to release neurotransmitters to communicate to another neuron, or if it's a motor neuron, to a muscle cell to make it contract. And so what happens at the end? And so that influx of sodium ions at that last node is going to trigger a series of events that is going to release neurotransmitters. So that's what we're going to look at. So looking at the bottom here, nerve impulse transmission between cells. So that's what we're looking at. How is it transmitted between cells? Nerve impulses travel between the axon of one neuron and the dendrite of the next neuron. Remember, the dendrites are what receives the stimulus. Or it can be between an axon and a muscle cell. It doesn't have to be another neuron. So it can be between one neuron and another neuron, or the postsynaptic cell, the cell receiving the signal, can be a muscle cell. And so there are two types of synapses. That space, so across what are called synapses, the space between the two cells that are communicating. Synapses are sometimes called synaptic clefts. There are, it says, at electrical impulses, or synapses, the electrical current flows from one neuron to another. So it's electricity that goes from one neuron to another. In most synapses are the second type, and that's what we're going to talk about here in class. At chemical synapses, a chemical neurotransmitter carries information across the gap junction or the space, the synaptic cleft. Most synapses, as I just said, are chemical synapses. So the presynaptic neuron, that's the neuron that's going to do the communicating, synthesizes and packages the neurotransmitter in what we call synaptic vesicles, located in the synaptic terminal. The action potential causes the release of the neurotransmitter, and then the neurotransmitter diffuses across the synaptic cleft and is received by the postsynaptic cell. So let's look at this in this picture. So we have two different types of cells in this picture. You have your presynaptic cell. So that's the one doing the communicating. So notice here, presynaptic cell, and it's going to send the signal down this way. Notice the arrow this way. And then it's going to communicate with the postsynaptic cell. So looking at this picture here, this right here is your presynaptic cell. So presynaptic membrane here. So that's this part right here. Alrighty, presynaptic membrane, that's the presynaptic cell. And then you have the membrane of the dendrites. Here's the membrane of the postsynaptic cell. So then you have your postsynaptic cell. Here, I'll do in a dark green here, postsynaptic cell membrane. Postsynaptic cell in green. All right, so those are your two cells. So now we're going to look at the communication between them. And so remember that synaptic cleft is that space. They're not actually touching the two cells. So this is the synaptic terminal, and this is the dendrite. And so they're going to communicate with one another by the presynaptic cell releasing a neurotransmitter. So here's your presynaptic cell. This arrow here represents the impulse from the node to node to node. So this, let's say, is the last node right here. And so just to illustrate that, let's pick a different color here. I'm going to do purple here. So your last sodium channel opens. Remember that the signal is moving down the axon. The last sodium gate opens, and so you have an influx of sodium. Sodium is going in, and the inside of the cell becomes positive or depolarized. All right, so so that's step number 1 here. So notice here the 1, let's write here, I'm going to write next to it. Step number 1, the sodium gates open at the last node. I'll put this in purple here, right over here. At the last node, sodium diffuses into the cell. And cell is depolarized. It becomes more positive. Now normally for the next node, that positive would cause the next sodium gate to open at the next node, but there is no other nodes. We're at the end. We're at the synaptic terminal. So that positive charge, what happens is it causes something else to happen. So notice here, step number 2, is that there's another type of channel. This is a voltage-gated calcium channel. Whenever we see voltage-gated, it means that the gate is shut and it only opens in response to a change in charge. And so let's write that here, that this voltage-gated calcium channel, I'm going to put here in parentheses behind here, only opens when neurotransmitter binds to it. Oh, sorry, not neurotransmitter. When a change in voltage occurs, a change in charge occurs. Got ahead of myself. The ligand-gated ion channels, I was looking at that. Those are when the neurotransmitters bind to it. So we'll get to that in a second. So the voltage-gated ion channels, the charge is picked up by that, and then the calcium, pick another color here, I'll do green for calcium here. So the calcium diffuses in when that channel opens. So we could write here for the next one, for step 2 here on this diagram here, is the calcium gated channels open in response to the depolarization from up here, the sodium gates openings from the sodium. All right, so then what happens is calcium diffuses into the cell. Where must the high calcium concentration be? Outside, right? So it diffuses in the cell, and the cell becomes even further depolarized. So the inside of the cell becomes even more positive. All right, so now notice what's going on here inside the cell is that you have something in the synaptic terminal that we haven't yet talked about, and that's these synaptic vesicles. These are vesicles that contain neurotransmitters. So I'm going to put in pink here the neurotransmitters. So neurotransmitters right here. So these are all these little vesicles making that the cell has made through the endomembrane system. So the Raphael, then the Golgi apparatus, remember those vesicles. So one thing that it could be that those vesicles contain after they butt off of that Golgi apparatus are neurotransmitters. So a neurotransmitter is what relays the signal from one neuron to the next. And so when we look at this, what's going to happen next is, you can see here, step 3 is those neurotransmitters, those synaptic vesicles with the neurotransmitter move towards the synaptic terminal and fuse. If you notice here, it's fusing with the cell membrane and releasing the neurotransmitters out into the synaptic cleft. And so that process is very similar to what process? Exocytosis. So So there's a lot going on for step 3 on this picture here. So we can write that the synaptic vesicles containing neurotransmitters move towards the synaptic terminal they fuse with the membrane they refuse with the membrane and do what? And what do they release? Release neurotransmitters into the, what do we call that space? Synaptic cleft. And release neurotransmitters into the synaptic cleft. Alrighty. And so that's what's going on here. So then, let's look here. What is going on with the neurotransmitters? What do they appear to be doing? Well, let's look at here. The postsynaptic cell has these ligand-gated ion channels. So voltage-gated ion channels, like this calcium channel, means that they're gated and they open and close due to a voltage change. So a ligand-gated ion channel means they're gated and they open and close depending upon if a ligand is bound to it. And so what we see here for these two, do you see these two neurotransmitters have bound to a special spot on these gated ion channels. And so, therefore, these are going to open due to that ligand binding to it, and the ligand happens to be the neurotransmitters. And so here, let's do another color. Let's see, what haven't I used? This blue. So these right here are your channels right here, and let's write the ligand-gated ion channels. These guys only open when what binds to it? When a neurotransmitter binds to it. So if we look here, if we look more specifically, they enlarge that so you can see it. So here is your ligand-gated ion channel. That gate is opened. Let's do the same color as the neurotransmitter. So the neurotransmitter is bound, and it allows it's showing you there's potassium gates and sodium gates, but what happens is the sodium is going to come in here, and the inside of the cell now will become more positive. So as sodium goes in, and the inside of the cell becomes more positive, so the inside, as those sodium comes in, so the inside depolarizes, which remember means it becomes more positive, which then can trigger another action potential in the postsynaptic cell. And so then it can, that can trigger the sodium gates at the first node to open, and the whole thing happens all over again. So let's write here for number 4, I'm going to do in this blue color here for number 4, the neurotransmitter binds to a receptor on the postsynaptic cell. Causing the sodium gates to open. Sodium diffuses into the cell. And the cell is depolarized. So therefore that cell has communicated with the next cell. And so if this is a neuron, then the sodium gates open at the first node, and so on and so forth, and it triggers the action potential to continue down the axon. So let's look here. So the response here at the end can be either stimulatory or inhibitory. Meaning that there are excitatory neurons. We looked at that when we looked at the knee-jerk reflex, where there's a stimulatory neuron that causes the muscle cell to contract, and there's inhibitory neurons that stops it from doing that, for the other muscle from contracting. That was this picture here. So you have stimulatory and inhibitory neurons. So it inhibits that and causes this muscle, the hamstring muscle, not to contract, whereas this causes it to contract. So there are stimulatory and inhibitory neurons, and how they work is stimulatory works just like we just talked about here. Inhibitory neurons usually hyperpolarizes a cell, so it makes it more negative, and therefore a stimulus can't make the neuron contract. So then what happens is we don't want the neurotransmitters to stay on here forever, because then you'd always have the sodium diffusing in. So after release, the neurotransmitter doesn't stay on, it detaches. It can diffuse out of the synaptic cleft and therefore not be used again. So they diffuse out of the synaptic cleft. It can be taken up by surrounding cells. So it can be taken in through endocytosis and reused. Or oftentimes it's degraded by enzymes. So there are enzymes that will degrade the neurotransmitter to neurotransmitters so that the cell doesn't continually get stimulated and this neuron keep on firing. All right, so what happens at the synaptic terminal? How do neurotransmitters work? So we just wrote here. So let's see. I would like you to pause it and see if you can kind of fill in the blanks here. See if you can do that about what happens. All right, so hopefully you paused it, and let's go over this. So really it starts right here in the synaptic terminal and not at the last node. So these sodium gates do open, but that's going to trigger the gates over here to open. So that's the calcium gates. So the calcium gates, if you don't know, Ca2 plus is calcium. Gates open. The synaptic vesicles release neurotransmitters. The neurotransmitter binds with postsynaptic, it didn't show here. It should there be a blank here. Postsynaptic receptors. That's this part here. The postsynaptic membrane is either excited or inhibited. That's whether or not it's an excitatory or an inhibitory neuron. And the excitatory causes sodium to come in, and the inhibitory causes potassium to go out. So that's why they're both here. It's a little confusing here. So when the potassium goes out, the inside of the cell, and sodium stays put. When the potassium goes out, the inside of the cell becomes even more negative, and that's what I was talking about with the hyperpolarized here. So that's how an inhibitory neuron, potassium goes out, excited, sodium goes in. And then eventually the neurotransmitter is degraded and or, I should put here, or recycled. A common neurotransmitter is called acetylcholine. Acetylcholine is a neurotransmitter that's released by motor neurons, and the effector cell is a cell of the skeletal muscles. So if this is a muscle cell here, this motor neuron will release acetylcholine, which then will bind to the muscle cell and trigger the muscle cell to contract. And so an example of an enzyme that breaks down that particular neurotransmitter is called acetylcholinesterase. And so notice the ACE for the enzyme. All right, and the last little bit are just to show you that there are different kinds of neurotransmitters. So there are more than 100 neurotransmitters belonging to 5 different groups. A single neurotransmitter may have more than a dozen different receptors in the various types of cells. So we just talked about acetylcholine. This is at the neuromuscular junction. So this is where, what does that mean? Neuro is neuron, muscular is muscle, so it involves muscle stimulation, memory formation, and learning. So it's where a neuron meets a muscle. So that's the one I just talked about. There's biogenic amines. Some examples of that is epinephrine. The epinephrine is involved in the fight or flight response. And can be derived from amino acids and are mostly secreted between neurons of the central nervous system. So what is the central nervous system? Brain and spinal cord. There are some that are just amino acids, so GABA. This actually acts as an inhibitory neurotransmitter in the brain. So it inhibits and therefore doesn't allow a signal to be transmitted. The other ones that we talked about were excitatory. There are Neuro Peptides. They're short chains of amino acids, can function as neurotransmitters. So substance P and endorphins both affect our perception of pain. Endorphins are kind of like a natural painkiller. So opiates bind to the same receptors. Just a little tidbit here. So opiates and opiate drugs bind to the same receptors as endorphins and therefore are used as painkillers. So we used our knowledge of natural painkillers, endorphins release and they bind and stop the transmission. These are also inhibitory because they stop the transmission down the neuron, and so therefore we don't feel it or sense it in our central nervous system as pain because it stops the signal from happening. And so we made artificial puts opiates artificially into our body that act the same and bind to the same receptors. And then some gases, nitric oxide and carbon monoxide, are also found in the peripheral nervous system as well. So I just wanted to show you that they could be gases as well. The big one to know here is the acetylcholine. Okay, but I just wanted to see that there are lots of different types of neurotransmitters that are released to be involved in communication. And that is the nervous system. We can continue, we could add our new information onto this. So the sodium comes in at the last node, causing depolarization. So see if you could, I would pause this and see if you can draw in what happens with this. So that positive charge is going to cause the calcium gates to open. So we can draw here, calcium gate open, calcium goes in. Inside of the cell becomes more depolarized as that calcium goes in. That's going to cause our special vesicles in here that have the neurotransmitters. Oh, I use pink for potassium. Let's do a different color. Let's do orange. It has the neurotransmitters inside, causes those to come over here and fuse with the membrane, releasing the neurotransmitters into the synaptic cleft. And then if you have, let's do the dendrite of the next neuron. So here's the dendrite of the next neuron. And so remember that you have sodium gates that are closed. Sodium gates that are closed. That neurotransmitter comes and diffuses, binds, causes that gate to open, and sodium diffuses in. Inside the cell becomes more positive. That's going to trigger then the first node, which is like this part here, just happened here. Sodium at the first node to open up, and the whole thing happens all over again. So that's how that relates to our big picture here. All right, and that is the end of the nervous system.